Sphingolipids. These sphingolipids have sphingosine in them and not glycerol. A polar head group is there, two non-polar tails are there, but no glycerol. So they are composed of one molecule of long chain amino alcohol, and that long chain amino alcohol is called sphingosine. Okay, sometimes its derivative also may be there, and one molecule of long chain fatty acid. And a long polar head group also is joined there. Okay. The carbon 1, 2 and 3 of the sphingosine molecule is just similar to the equivalent carbons of glycerol. We will see them in the next slides when we see the diagrams. Ceramide is an example of a sphingolipid. In ceramide, a fatty acid is attached in amide linkage to the NH2 on C2 of the sphingosine. Ceramide. Ceramide is a structural component in all sphingolipids. Remember, a fatty acid is attached to the carbon 2 of the sphingolipid or sphingosine in amide linkage. Ceramide is basic figure. Just like, in the previous slides, you found that in the glycerophospholipid, phosphatidic acid is there. Like that, in this sphingolipids, ceramide is there. There are three classes of sphingolipids: sphingomyelins, neutral uncharged glycolipids, and gangliosides. You see, this is the general structure of sphingolipid. Here, this is the sphingosine. Sphingosine is this color, CH2, carbon number 1, carbon number 2, carbon number 3. This is the sphingosine molecule. This is the amide linkage. The fatty acid is actually there in amide linkage. This is carbon number 1. This X is a variable group. What is that variable group? Accordingly, different sphingolipids are there. The X is merely a hydrogen atom. We call it a ceramide, basically present in all sphingolipids. And when it is a phosphocholine, when the X is a phosphocholine, we call it sphingomyelin. Keep it in your mind, we will see it later. When this X is a glucose, is a good example of a neutral glycolipid or glycosyl cerebroside. When it's an oligosaccharide, we say it is a lactosyl ceramide. Like that, there are different types of sphingolipids. Coming to sphingomyelins, what's a sphingomyelin? We found just now in the previous slide, we found what's a sphingomyelin. Contain a phosphocholine or phosphoethanolamine as their polar head group. You see, this is the sphingosine. Here is the fatty acid. Here there is the phosphate group attached. Onto the phosphate group is a choline attached. Here can you see a positive group? So this positive group make this region a polar head region. And here there is a negative charge as well. There is a polar head region. This is a typical example for a sphingomyelin. They are present in the plasma membranes of animals, and they are especially prominent in the myelin sheath around neurons. Okay, now you might have understood why do we call it myelin sheath. And we have the glycosphingolipid. In glycosphingolipids, carbohydrate is present as we found in the previous slide. You can see here, this is the sphingosine unit, this is the fatty acid, and here you have the carbohydrate, glycosphingolipid. And gangliosides, here we have N-acetyl neuraminic acid, or simply we say acyanic acid in place of the carbohydrate. And why we are so much concerned about this sphingolipids, because it is there in our body. You might have heard of blood group antigens. Some of you may have antigen A in your blood. Some of you may have antigen B. Sometimes you may have both antigen A and B, and sometimes you may not have antigens. Then we call you O group people. What does it mean, A group and B group, etc.? See, what you see here is the structure of O antigen. What is O antigen? Remember, these antigens are present on the surface of RBC. What's in O antigen? Sphingosine and fatty acid. Basically, ceramide is there. Then, on the third carbon, there is an oligosaccharide. Glucose, galactose, N-acetyl galactosamine, galactose and fucose. You number them. 1, 2, 3, 4, 5. Which are they? Glucose, galactose, N-acetyl galactosamine, galactose and fucose. This is common to all the blood groups mentioned here. Okay. To this, if an N-acetyl galactosamine is added, you see already we have a galactose here. This galactose is this galactose. To this galactose, if an N-acetyl galactosamine is added, that is antigen A. If you have that sugar on your lipids, on the plasma membrane of the RBC, we say you have A blood group. In that place, instead of N-acetyl galactosamine, if a galactose is added, then we say you have antigen B, you have blood group B. So this is the only difference between antigen A and antigen B. When N-acetyl galactosamine is there added to a galactose, then it is antigen A. When galactose is added, you call it antigen B. When both these are added, we call it AB blood group. Is it clear? That much important are the sphingolipids. Then coming to steroids, sterols. You might have heard of there's an error in this one. This is not OC. It is actually a bond, CC bond. You read like this: C bond C, carbon-carbon bond. That's the meaning, not O. Okay. Sterols are structural lipids present in the membrane of eukaryotic cells. They have a steroid nucleus, and we will see the structure of the steroid nucleus. One of the most important steroid is cholesterol. Cholesterol actually consists of 4 rings, and 27 carbon atoms are also there. From the cholesterol, many hormones are formed in our body. Bile acids and bile salts are formed in the body. So without cholesterol, we cannot lead a normal life. And you might have heard of atherosclerosis, etc. There this cholesterol in association with some proteins are accumulating in your blood vessels. That's why it is leading to atherosclerosis and cardiac arrest. So it is very much essential for our survival. If the amount is too much, we are in trouble. If the amount is too little, again we are in trouble. This is cholesterol. See, there is a ring number 1, 6-membered ring. Ring number 2, again 6-membered. Ring number 3, again 6-membered. But ring number 4 is a 5-membered ring. Can you see? And then this is the side branching. You should know how to draw it. When we come to the actual class, I will show you on the board how to draw it. Okay. This is the cholesterol. You see, there is a characteristic ring system present. It is called steroid ring or cyclopentanoperhydrophenandrine. This ring is called cyclopentanoperhydrophenandrine ring system. Okay. You see, this is bile acid. Bile acid is derived from cholesterol. You see, taurocholic acid. This is testosterone, a male sex hormone, is derived from cholesterol. And they are membrane constituents. These sterols are sometimes constituents of membrane. They are regulating the fluidity of many types of membranes. So essential for our survival. Ergosterol is actually a plant, not a plant, and there's a fungal steroid. Basically, it is found in ergot fungus. That is why we call it ergosterol. Okay, and they are also present in protozoa. Why they are important, you know? You know that sometimes fungi, protozoa, etc. are causing diseases in our body. Many of the medicines we use against these organisms are targeting their ergosterol. See, many fungi and protozoa are having ergosterol as an essential component of their body, their cell, and we have medicines targeting this ergosterol present in their cell. That's why clinically they are very much important. And you know, this ergosterol is the source of vitamin. It's a pro-vitamin actually for us. It's actually pro-vitamin form of vitamin D2. From this ergosterol, vitamin can be produced. I mean, there can be formed in the body when the body is exposed to UV light. So essential for our survival. Only this much in this chapter.